TET systems, or Tetracycline controlled expression systems, come in two forms: the inducible Tet-ON system and the repressible tet-OFF system. And in this video, we shall discuss both of them. The TET system is based on this naturally occurring tetracycline operon that ensures resistance against the tetracycline antibiotic. This operon is found in a transposable element in the bacteria. I'm not going into details of the operon, but here's what you need to know. There are quite a few operon-specific genes that help in antibiotic resistance. The important regulator for all these genes is the tetR gene, which, as the name suggests, produces a repressor protein called tetR. The tet repressor specifically binds to tet-O, the operator sequence, which is 19 base pairs in length. In the default state, when no tetracycline is present, you don't need to waste energy making products from these Tet Operon genes. And this is what happens. The tetR binding to the operator keeps the operon off. But when tetracycline shows up in the environment of E. coli, tetracycline binds to this repressor and allosterically inactivates it. Now, the tetR cannot bind the operator. The genes are now freely transcribed and proteins from this Tet Operon are made. So, when tetracycline is around, bacteria needs resistance, and allosteric inhibition makes sure Tet Operon is activated. A word of caution: do not confuse the mechanics of the operon on and off with the tet-on, tet-off. As you will see later, they're very different. Okay, now let's think about using the components of this operon. This tet-O, a 19 base pair region, specifically recruits the repressor. This is functionally the same as the 17 base pair lac operator where lac-I is involved. Essentially, the repressor is a DNA binding protein, and that is all it can do. It does not have any other activity. It is not a transcription factor. It just binds DNA. That's it. But TET systems are used to control gene expression. So is there a way to transform this tetR to behave like a transcription factor? Well, there is one way. The transformation comes with the help of a VP16 protein from the herpes simplex virus. VP16 is a small little protein that has a core and an activation domain in its C-terminal region. The activation domain is strongly acidic and has a net negative charge, just like most eukaryotic transcription activators. The core, on the other hand, seems to have some DNA binding ability. The activation domain, because it is similar to transcriptional activators, binds a lot of eukaryotic factors like TF2B, TBP, TF2D, and a lot more. Therefore, unsurprisingly, the activation domain can attract RNA polymerases. Some small side note: the activation domain is the last 80 to 130 amino acids of VP16, depending on the paper that you read from the 90s. But the acidic patch of about 12 amino acids is what is being used today in the TET system, at least in the one sold by Takara. Now, given that tetR binds DNA, we do not need the DNA binding capability of VP16. It is not that good anyways. But instead, we take the VP16 activation domain and fuse it at the C-terminal end of the tet repressor. This fusion protein is called tTA, which stands for Tetracycline Controlled Transactivator. And here's a cute little picture of it. The original papers added a single activation domain to the repressor, but Takara's version has 3 repeats of this 12 amino acid for a stronger effect. In case you're curious, here's the tTA from Takara with tetR in the front and acidic patch repeated 3 times at the C-terminus. Now, this tTA can bind an operator, and if this operator is placed behind a promoter, the activation domain will recruit RNA polymerase for transcription. So we have taken tetR, added an activation domain to it, and converted the DNA binding protein into a functional transcription factor. Great. Now let's start our discussion on the tet-OFF system. A simple way to deconstruct this is to read it as when tetracycline is present, your protein is off. By the way, oftentimes you use tetracycline analogs like doxycycline to prevent cellular stress from the antibiotic. In a typical tet-OFF system, you have a synthetic promoter called the TRE, which stands for tetracycline responsive element, At least the version from Takara has 7 copies of the same operator in tandem, followed by a minimal promoter, which is generally the CMV promoter or the EF1 alpha. These are designed to be minimal promoter. They often contain mutations, so they do not have significant levels of activity on their own. The repetitive tet-O ensures that a lot of RNA polymerase can be recruited at the minimal promoter. In front of the response element is where you clone your gene of interest. In a scenario where you do not have a tetracycline or DOX in the system, the repressor part of the tTA will bind to the operator. As we just discussed, the activation domains attract the transcription machinery to this particular location, and from the minimal promoter, you start expressing your gene of interest. In contrast, where you provide the TET or DOX, the tetracycline binds the repressor, inactivates it, and the tTA then no longer binds to the operator, which means no transcription and no protein production. Here's an intuitive look at the usage of this system. There are 2 parts to this. One is that you must have a gene cloned next to the TRE. Second part is where you have the gene for tTA being expressed. These could be permanent cell lines, mouse lines with specific promoters expressing the tTA, or tissue-specific promoters, and you control the expression of your gene through DOX or TET treatment. A classic way is perhaps through plasmids, which of course is sold by Takara. One plasmid contains the TRE, where you clone your gene. The second plasmid contains the transactivator. This is Tet-OFF 3G, and you co-transfect both of these plasmid to get transient expression control system instead of a permanent one. There is also a single plasmid version of it called Tet-ONE, which again is a plasmid-based system for transient expression. There is one concern with TET systems in general. It doesn't matter if the tTA is active or inactive. During inactivation, only the repressor part is affected. VP16 domain is always in an active state. So regardless of active or inactive state, the tTA still attracts RNA polymerase to itself. VP16 is very powerful. So higher the tTA expression, lower the amount of transcriptional machinery that is available for cells to do their job. This can lead to cell death. This protein titrating behavior of tTA is called squelching, and you may have to adjust the dynamics and regulation of tTA promoter in case you notice your cells dying. The counterpart to tet-OFF is tet-ON. When tetracycline is present, your protein is on. However, the Tet-ON does not use the same tTA as we have discussed so far. Back in late 90s, the TET repressor was mutagenized, and a few repressor variants with 4 to 5 mutations in its binding domain were discovered. This variant repressor is called reverse tTA because it does not bind the operator in its default state, thanks to the mutations. This means, in the scenario without DOX, the reverse transactivator cannot bind the operator, so you do not see your gene being expressed. But when you add DOX to the system, it binds rtTA, changing its conformation such that it binds to the operator. This reversion of rtTA is allosteric activation, as opposed to the allosteric inhibition of the tTA. So, with tetracycline in the Tet-ON system, you expect to see the gene expression. Choosing between on-off system in practice depends on your application. That is, whether you want your gene to be expressed by default or not. And just like TET system depends on tetracycline and repressor combination and VP16 fusion, there are a range of similarly regulated arrangements based on LAC-I, GAL4, and many more. I hope this video was useful, and I will catch you in the next one.